there is a lot of euphoria, excitement, but there are challenges. I think we are all well aware of them. Now, I am personally optimistic from what I have seen happening in vector development in the past 5 to 10 years. I know now that there is no challenge that we won't be able to meet. Now, we may need to tweak a little bit what we want this vector to do or what we want the treatment to be in a human being, right? So, in the terms of challenges, I'm probably going to go through 3 categories. The first and foremost, surprisingly, for some, it is still production. It is still manufacturing large scales for clinical and for commercial, right? It has happened. There are 2 AAV gene therapy, 3 actually, marketed products. So it is possible, but it is still a struggle. So with the amount of effort and funding that is now being invested in platforms, it's happening, but it's not resolved. So manufacturing, production, a large amount is one challenge. In the same line of thought, the purification. So you're producing a huge batch. You have to purify these vectors to a level of purity that is extremely, extremely high to meet with safety concerns and also potency concerns. So there is a lot of room for improvement in the purification of these vectors. One of the complexity of this drug, which is very different than when you talk about aspirin or ibuprofen, is that each of the products, even if they are all AAV, each of the product is almost a different product. And you may have a different way of purifying this product. And so a universal platform for the upstream or for the downstreams hasn't really been developed with the idea of applying to everything. So that's why there is a lot of vector development still. And the third, it's kind of 3 and 4 challenges, the third and the fourth at the same time, but they are combined. It's obviously the safety of these drugs, and I'm going to link that to the level of residual and impurities that are part of these lots. We've always considered AAV to be a very, very safe vectors compared to adenoviruses or lentiviruses or retroviruses. AAV is a very safe virus. Just the wild type virus is very safe. We are, most of us, infected with it. And the vector, until very recently, had been tested in vivo in human and be proven to be very safe. What has happened, though, was with the progress made in production manufacturing and in scales, and trying to tackle some inherited diseases like Duchenne muscular dystrophy or other myopathies, the demand for the high doses in human has really grown up, and we were able to produce them. So higher doses have been administered in the past year or 2, and we have seen serious side effects. Now, do we know really what was the reason? I think this is still being debated. Is it the dose in the vector itself, or is it the amount of residual impurities that you are bringing in? So when now everything gets connected, I always like to say is that if you keep doing a good vector development to improve your production, yield, titer, improve your purification to reduce the residual, and improve the potency of your vector, you're going to end up having to hopefully inject less of this vector. And so it's a positive feedback loop where vector development is feeding into safety, safety drives vector development, and so forth. So continuum of progresses that will tackle these challenges, but still a lot of work to do.